Hello again. Today's lecture will cover the forensic examination of hair and of fibers. We'll start with hair. First, let's clear up a few common misconceptions about hair. Hair is a mammalian feature. In other words, it occurs in mammals. There are somewhat similar things in other animals that may be called hairs, and also in plants, as we talked about cystolithic hairs in marijuana. But actual hairs are a mammalian material. So the hair grows from pores in the skin called follicles. So the follicle is the place where the hair initiates and grows, not the tip or the base of the hair itself. The follicle is separate from the hair. It's simply a place where the reproduction of a new hair and the growth of a hair take place. Hair grows from the base, from the follicle, upward, so no hair grows from the tip. Most plants do grow from the tip, so if you cut off the tip, you're cutting off a live part of the hair of the plant. But if you cut off the tip of a hair, you're not cutting off any live part, because the hair is not a living organism or is not alive, has no pain, has no growth, has no connection really with your body. You don't feed it with your blood. You don't feel any pain if someone cuts your hair, because it's dead tissue. So oftentimes you hear about someone's hair turning white overnight. No, that isn't likely, nor is it thought to be possible, because there's not a mechanism that connects your brain to your dead portions of your hair. Another misconception is that hair continues for a long period of time. Actually, each follicle grows for a set length of time. Human head hair, for example, grows for somewhere a little bit less on average than 2,000 days. At the end of that time, the hair stops growing, goes dormant, and shortly thereafter falls out. So if you find hair in the bathtub or on your comb or hairbrush, don't get alarmed. That's the natural process. There's always a certain number of hairs that are in the dormant stage waiting to fall out. And they will fall out naturally. It doesn't mean you're going to go bald. Which reminds me, you may find it a little ironic that I am giving a lecture on hair when I don't have a lot of experience with it. But I certainly have a lot of experience with the forensic examination of hair. You cannot speed up or slow down the rate of growth by cutting or by trimming or by shampooing or by any of a number of what I call snake oil preparations. There are some medicines now which will stimulate the growth of hair, but not to make it grow any faster necessarily or for a longer period of time. That being said, let's talk a little bit about the hair. The hair you can kind of compare it to a pencil. It has 3 layers. The pencil has paint, wood, graphite. Not lead, graphite. So hair has 3 layers. The outer layer, which is a very thin layer consisting of only a few layers of cells, is called the cuticle. The cuticle is a protective coating primarily. That is where the surface is protected by the layer of cells which we call scales, and they kind of look like scales, but they're not like fish scales that you can scrape off easily. They're very much a part of the hair, and different animals have different shapes of scales, and that's one of the characteristics that we use in the identification or examination of hair. The middle layer, which in humans represents about 2/3 of the the bulk of the hair, is called the cortex, and that is where the pigment granules are. And pigment granules are what give the hair its natural color. The more pigment granules you have, the deeper is the color. The primary pigment in humans is melanin, and that accounts for the brown to dark to black hair. So the more pigment granules you have, the blacker your hair is. And the pigment granules may occur in bunches or in streaks. They may be dense, they may be widespread. They're quite small, but you can visualize them under regular light microscope. And they again are one of the characteristics we look at when we're examining hairs. The center core in humans, about 1/3 of the diameter or less of the hair, is called the medulla, which means middle. So it's the middle of the hair, and it is as the hair is growing, it consists of a fluid. But as the hair emerges and grows out a little bit, it dries out, and the medulla becomes air-filled cells. And when you look at them under the microscope, the medulla looks like a black line. It's black because it has a different refractive index than the area around it. So it's the contrast is the medulla, and the medullas have different shapes, different widths, and different characteristics, primarily in non-human hairs. Sometimes the medulla can be very characteristic in identifying the species of a hair. So that's the basic makeup of the hair. It is it grows, incidentally, head hair that is, grows at approximately 1 centimeter a month. So you can kind of figure maybe around 1/2 inch a month growth in the normal active head hair. The root or base of the hair, which is in humans kind of like the bottom of an onion, that is, it has a little bulb on the end, and that is what resides in the follicle and is the mechanism for reproduction of hair cells and makes the hair grow. The tissue surrounding the root and adhering to the root while it grows is important in some forensic examinations. The growing process is, as I mentioned earlier, separated into 3 stages. The first stage is the anagen stage. That is the growing stage. The follicle is attached to the hair root, and the hair is actively producing cells and getting longer. The second is the catagen stage. That is the time in which it is growing dormant. The growth processes are slowing down, and the connection between the root and the follicle is diminishing. The blood, which has previously nourished the root, separates, stops coming, and the hair prepares to become completely dormant. The dormant stage doesn't last very long, and the third stage is the telogen phase. That is when the hair is dormant but still loosely attached to the follicle. If you pull the hair, it will easily come out, and the base, the root, will be clean and free of cells. So we can look at a hair and determine what stage the growth is in that hair. Sometimes that's important. Sometimes if you're attempting to show that a particular hair was forcefully removed, as in a physical confrontation or a fight, or maybe being struck with a tool, then if you can find follicular cells attached to the root, you can assume that it was removed forcibly. I recall one homicide in Ogden where they found a man beaten to death in an alley, and we didn't have a suspect, we didn't have witnesses. They found not far from him, not far from his body, a 2x4, and there was blood on the 2x4. And when I examined it, I found hair consistent with the hair of the victim, which had follicular cells attached to some of the roots. So I concluded that that was the weapon that was used to bludgeon him to death. Another case where that was of interest was the case in which two men got into an argument, and in the course of that argument, one of the men was killed. He had numerous bruises and cuts on his face, including some right around his eyebrow. And it was surmised by the law enforcement, the prosecution, that he'd been struck with an object. They found nearby a rock about the size of a little bigger than a softball, and there was blood on the rock. The defendant in the case said that he had never used a rock, he knew nothing about the rock. When I examined the rock, I found blood and also hairs. When I examined the hairs, they were eyebrow hairs, and so we knew that they had come from someone's face, and they also had follicular tissue on some of them, so we knew that they had come out forcefully. So they were able to prosecute for homicide, and the weapon being a rock. So we concluded that the man had been beaten to death with a rock. Another very, very important part of follicular tissue is that it is fresh and growing, and therefore contains DNA. Hair is a series of cells, so hair contains DNA, but the shaft, that is the main part of the hair, is very difficult to analyze. It's very hard to break up, either chemically or physically, and the DNA that's extracted from the shaft of the hair rarely gives us sufficient results for an identification. The root, particularly if it contains follicular cells, often does contain enough nuclear DNA for an identification. So in that regard, it becomes really valuable to find hair that's growing and has cells around the root, which we call follicular, from the follicle of the hair. Here is a chart that shows the steps in the sequential procedure of examining a hair. The first step is to determine if it's a hair, and you can do that quite quickly under a stereoscopic microscope at low power, just to see the morphological features of a hair. Second, to determine whether it's human or non-human. The books all say it's relatively easy, but I think those books must be written by people who haven't had a lot of experience with non-human hairs. Certainly there are things about human hair that are unique, but they're degrees, not necessarily steps that can be easily recognized. Some hairs are easily eliminated from being human on the basis of color or unusual non-human-like medulla formations or scale formations. But there are a couple. Yak hair, for example, is quite similar to human. Some dog hairs can be confused, and occasionally some cattle hair is quite similar to humans. In any event, that's the first thing to do, is to determine whether it is in fact human hair or not non-human hair. Here are some photos of various medullary configurations. The first one on your left is from a rabbit, and that is quite characteristic of rabbits and hares, that multiple ladder effect. The second one is a deer. Notice that the medulla on the rabbit and on the deer are very wide. In the deer, it occupies almost the entire hair, with just a small amount of cortex. Both of those, however, are general characteristics of a group. You can't determine the species, only the group that they're in. For instance, the deer wouldn't be distinguished from another type of deer, such as a whitetail or elk or antelope, pronghorn antelope. So the next one on moving from left to right is a dog, and you can see there's some major difference in the medulla of the dog and the human. The human is just a black line in this case, whereas the dog is broken up into segments. Not all dogs look like that, and not all humans have an unbroken medulla. In fact, some human hairs don't have a medulla at all. So you can see that medulla and the internal characteristics of hairs are helpful in determining species, but don't necessarily give you all the information you need. In the examination, if it's determined to be non-human, sometimes that's all we need to know. Sometimes we need to know what species it is. For example, I had a situation where an animal was killed in the zoo in Salt Lake City, and the examination of the area found a hole in the fence, and caught on the fence on the barbed wire of the fence was a small tuft of hair, and the authorities were very interested in determining what kind of hair that was, because they wanted to know what kind of animal had killed the antelope in the zoo. So I had to examine that hair and determined that it was in fact domestic dog hair, and they then decided it must have been someone's dog, either out running around loose, or maybe even gone wild. But we needed to know if it had been another type of animal, maybe they would have taken some precautions in addition to what they did to keep animals out. I also have done a number of other animal hair cases. One that I remember was a what's called a canned cougar hunt. Mountain lions, or cougars, are game animals in Utah, and you can purchase a license to hunt cougars. There are lots of restrictions and numbers and so forth, but there are a lot of hunters who think it would be really great to shoot a mountain lion. So the Division of Wildlife Resources was visited by a man who had been down to the central part of Utah hunting mountain lions. He had a legal license, and he had in fact killed a lion. But he was a little disturbed when he thought about it, because it just didn't quite seem right to him. He thought, that was an amazing coincidence. It seems that he had several days set aside, and he hired a person to guide him, and they hunted and hunted and hunted and couldn't find any lions. And finally, on the last day of his trip, he had to go home the next day. They took him to a new place, and as they were driving down the road looking for cougar tracks, they made a stop for relief, and all of a sudden one of the guys said, "Look, there's a lion," and a mountain lion was trotting across the road. So the hunter grabbed his gun and shot the lion, and he was happy as could be. But when he thought about it, he thought, no, that's just too much of a coincidence, that it would be the last day and we would be on a road and we'd just stop in the very right place and get the lion. So the division officers proceeded to investigate that. They went to the home of the guide, and they noticed in the backyard a large pen, which got their interest. So they went to the pen, and there were hairs on the inside of the pen, which they collected and brought to me, and I determined that they were mountain lion hairs. It seems that there are unscrupulous mountain lion guides who capture a lion, which seems like a very difficult task, and I don't think I want to try it, but once they get the mountain lion up in a tree, I guess they can get a rope around it or somehow capture it, and then they just hang on to it until they have a client who wants to shoot one, and then they turn it loose at the right time, and he gets to shoot it. And that's illegal. So they charged the guide with that crime, and I testified that there was in fact mountain lion hair, and I believe they convicted him. So there's lots of applications for determining the species of hare, and they're what we might think of as domestic hares, dogs, cats, hair from clothing, mink coats, rabbit fur, gloves, and so forth. So most crime laboratories don't specialize in hair, and so many of them are not able to identify beyond the common domestic hares. I suspect if you're in New York City, you want to know how to identify cat hair and dog hair, and maybe a few others. But it wouldn't be like out here in the Wild West, where you might get coyote hair or mountain lion hair or any kind of deer or elk or different kinds of rabbits. We had a case in which a Division of Wildlife officer went to check a couple of guys who were, he could see them from quite a distance walking through the snow in Parley's Canyon. So he went over to check them, and when they got to the road, he asked them what they were hunting. They said they were hunting snowshoe hares. That's the type of hare that changes color. They're kind of a grayish color in the summer, and in the winter they turn white. There's a season for those, and the season was not on at the time. So the officer informed them that, in fact, it's a good thing they didn't have any because it wasn't in season. So they left, and he got suspicious, and so he kind of backtracked them a ways, and about 30 or 40 feet from the road, he found some dead snowshoe hares. So he went back, jumped in his truck, and sped down the road and actually caught up with them and pulled them over. And when they got out, he noticed some hair attached to the shirt pocket of one of the individuals, which he removed and brought to me. And I determined that it was, in fact, from a white hare, as in rabbit, hare, H-A-R-E. So when I went to court on that one, the prosecutor said, "Mr. Gaskell, do you recognize this?" "Yes, I do." "What is it?" It's a hare. And the prosecutor said, "Well, can you identify the species?" And I said, "Yes." He said, "Okay, what is it?" And I said, "It's hare." One of my favorite jokes. Hare. Actually, that's a true story, though. In any event, there are a number of publications that you can use to help you identify animal hairs. One of my favorites is the publication by the Wyoming Game and Fish, which is the dorsal hairs of various mammals of Wyoming. It has a nice key in it, and I've used it many times to identify hairs of various kinds. If you determine that it is in fact human hair, and assuming you have a question sample as well as a known sample, you can proceed to make a comparison. We do have hair comparison microscopes, which are different than firearms comparison microscopes. They are, in fact, higher magnification and regular compound microscopes that are bridged together in such a way that you can put two samples under the scope at the same time and manipulate them and line them up side by side to see if their various characteristics are the same. Are they the same diameter? Are they the same medulla, same scale pattern, et cetera, et cetera. So having completed that comparison, if they fall within the range of each other, it's not one of those questions where all the hairs of a human head are not the same by any means. So you have a range. It may be a range from light brown to dark brown. It may be a range from wide medulla to narrow medulla. So you take all those characteristics and you say, okay, these two hairs are in the range on all of those characteristics, and therefore I can conclude that they could have been from the same person. Emphasis on could have been. Never in my career have I ever said microscopically these two hairs are the same, so they must have come from the same person. Hair is not like fingerprints. It doesn't individualize. It gives you a range, and you say, okay, these hairs are in the range, could have come from. And then you have to rely on your experience to make some kind of determination as to the likelihood that they came from the same person. And there are hairs with very distinctive characteristics. I had a homicide in Wyoming, and the suspect's hair was unique. I had never seen hair like that before, and I haven't seen any since. And that's what I told the court. I said, this is a rare hair. There are characteristics of this hair that are unusual, and I've looked at thousands of hairs. And so, my opinion is that there's a pretty good likelihood that these two hairs are from the same person. But that's as far as I would go. Once we had a case in Utah County where an elderly man was beaten to death in his home. And when the police got there, they found a hat at the scene of the crime, a baseball cap, and they subsequently developed a suspect who was over in Nevada using the victim's credit card. So there were eyewitnesses who could say, yeah, he owns a hat like that, and at least one said, I saw him wearing that hat recently. So they brought the hat to me, and I found in the hat several human hairs and several dog hairs. They went to the suspect's home, collected a few of the dog hairs and a few of the suspect's hair, and brought them to me. And in fact, the human hairs were within the range of his hair sample, and the dog hairs were in the range of his dog's hairs sample. And so I testified, and I actually gave a number. They said, what's the likelihood of two people having these same matching characteristics? And I gave them a number, and they convicted the man. Subsequently, that case was appealed to the Supreme Court, and one of the basis of appeals was that they shouldn't have allowed my number to be used because, well, we didn't have Daubert in those days. It was before Daubert. But the same thing applied. They said, well, there's not enough evidence to support Mr. Gaskell's using that number. The Supreme Court actually ruled that in this case, the use of that number was acceptable. So I may be the only person in Utah who has had a Supreme Court decision upheld when it comes to hair. But that decision was upheld. They denied the appeal. So getting back on track a little bit, I guess I should mention that dog hair and cat hair, we look at enough of those, and there's enough variety in dogs and cats that we can differentiate between individuals of the same species. Not that we can say absolutely this was from a dog, but we can say that these two dog hairs, came from dogs which were the same color and had the other internal characteristics of their hair. I once had a student who worked for a veterinary clinic, and I asked him if he could get me some dog hair samples, which he said he could, and he brought in, before the semester was over, samples from 50 different dogs, which I then sat down and compared side by side all of the hairs to each other, and there were 4 out of the 50 that I couldn't distinguish. The rest of them I could distinguish from each other. So if that's a representative sample, which I'm not claiming that it necessarily was, but if that is a representative sample, then 4 out of 50, that's 92% of the time I can distinguish dog hairs. And I don't use that number, and I don't think I ever would. But I will say that if dog hairs or cat hairs are what we would call a match, which I've already defined, that they're within the same range, that is significant. We had a man who broke into a house in Weber County and attacked a woman, and unbeknownst to him, that woman was in tip-top physical shape. She actually taught athletics. She was a sports teacher, and she proceeded to beat him up. She was tougher than he was, which I think, well, that's great. That made me happy. But in any event, she called the police, and they came, and the only thing she was sad about was that he escaped. So subsequently, a few hours later, another report came in of another girl who'd been assaulted, And when they went, they got basically the same description. And they subsequently developed a suspect. And in the course of things, they brought me hair samples from the first girl's house, from the second girl's house, and a shirt that the suspect had on. Turned out all three, they were cat hairs, and turned out all three of the samples were consistent. So I made up a report to that effect, and the defense attorneys immediately said, well, that's ridiculous. The suspect owns 4 cats. It's probably one of those. And we said, well, bring them in and we'll check it out. Much to their dismay, they brought in samples from the 4 cats belonging to the suspect, all of which were easily distinguished from the hair which was on from the scenes of the crimes. And they were sorry they'd brought the issue up after that. But in any event, we were able to link the 2 crimes together by cat hair. So, okay, so that we need to remember that it's not a fingerprint. Now, the FBI, for many years, had testified that a hair match was virtually the same as a fingerprint. About 15 years ago, they got in some significant trouble for saying that, and they've since changed and said no, it's just a probability, and it's not identical. It's not an individual characteristic. In the meantime, the hair comparisons have lost a little of their appeal, their luster, you might say, and some laboratories have decided not to do them just because of that situation. To me, that's like throwing out the baby with the bathwater. You should continue to do them, but just report them accurately. Don't say they're individual matches. Say that they're consistent with, they could have come from the same person, and then proceed on to do additional testing. So a couple other things we can learn from examination of hair. The hair on most mammals is not the same all over the body. Maybe change due to seasons as well. So oftentimes animals change the color of their hair or the consistency of their hair during winter. Some animals get a heavier coat. Some animals, when they get the heavier coat, get a more cold resistant hair layer. Humans don't change their hair in the different seasons, but we do have different kinds of hair, or different types of hair, if you will, on different parts of our body. And most of you, I'm sure, are aware of this, but maybe hadn't thought about it. Turns out we can identify scalp hair, facial hair, eye hair, pubic hair, and body hair. By body hair, I mean the hair on your shoulders and chest and arms and legs. There are some hair specialists who claim to be able to tell the difference, some differences in body hair, but I've not found that to be something that I do. I just call it body hair. So the differences in those types that I've mentioned are mostly the diameter of the hair, the stiffness of the hair, the length of the hair, and the cross-sectional shape of the hair. But it, of course, is good to know. Now, you should know that if we're making a comparison, so we have a head hair sample, a scalp hair sample, we must compare it to another scalp hair sample. We can't compare that to a beard sample, and so forth. So it has to be the same type of hair to make a comparison. So let's talk about a couple other things that we can or cannot do with hair. When I was young, it was really easy to tell whether the hair was from a man or a woman, because men had short hair and women had long hair. And of course, that's gone out the window, and now men have short or long hair and women have short or long hair, and the length is not particularly important at all. Although if you're looking for a suspect and you have a hair that's 12 inches long, then you can say, well, looking for someone with hair that's 12 inches long at least. But another thing is that people ask about is racial origins. And it's true that there are some general guidelines that distinguish Negroid hair from Mongolian hair or Mongoloid hair versus Caucasian hair. And occasionally when we get a hair and we see that those characteristics or those features are straightforward, we might suggest to an investigator what racial group he's looking for, but they're so inconsistent and so many crossovers and intermediates that really it's not a thing we can do on a case-by-case basis. And another thing that we asked about is age. Infants and fetuses have a hair which is very fine and short and blunt-tipped, which would distinguish that type of hair, that age of hair. But once they are born and they begin to grow their hair, it becomes impossible to determine age on the basis of hair. So to recap, in human hair we can determine if it's human, we can determine the color, we can determine numerous morphological characteristics microscopically, but at this point we cannot microscopically individualize hair and say it came from an individual person. We're able to do that sometimes from DNA. As we mentioned a little bit earlier, DNA is present in low levels in the shaft of hair, but oftentimes in sufficient levels in the root of growing hair. So if we have a growing hair root, particularly if it has follicular cells attached to it, we can oftentimes get a DNA profile. There is another type of DNA which is not in the nucleus, but is in the mitochondria, and that is mitochondrial DNA. And there are many, many more copies of that per cell. In the nucleus, there's only one copy of the DNA, but in the mitochondria, there may be hundreds. And so we can quite frequently get mitochondrial DNA information. It's an entirely different type of DNA. It is not as nuclear DNA is. It is not individual. So nuclear or mitochondrial DNA is passed on from mother to children. So all of your DNA, you got your mitochondrial DNA you got from your mother. All of your mitochondria you got from your mother. And so if you have siblings, or if you have cousins whose mothers are the same as your mother, that is our sisters, then they will have the same mitochondrial DNA. All of you, you and your siblings and your cousins and so forth. So mitochondrial DNA is not individual. It's a type of DNA that, of course, there are differences, there are mutations, and we can do it, although not all labs do mitochondrial DNA. But the odds of two people having the same DNA are not really well established, but are maybe in the neighborhood of 1 in 500,000, or even down as low as 1 in 30 or 40,000. So mitochondrial DNA is not bad, but it's not nuclear DNA, and therefore sometimes we can do individualizing of hair, and sometimes we can't, based on the characteristics of the hair. Now a little bit about collection and preservation of hair. Of course, hair at the scenes of crimes can be very, very important, and we need to collect it whenever we can find it. It requires tedious searching, and oftentimes with the use of a magnifying glass and a good light. But remember, we also need to follow the regular rules in collecting. Where was it found? When was it found? Who was there? Who saw you collect it? And what are the environmental conditions? Was it really windy if it's outside? And so forth and so forth. So normally when we find a hair, we can just pick it up with forceps or tweezers or other tools. But another way to collect it is by taking a roll of fingerprint lifting tape and forming it around your hand backwards, so that you end up with a circle of sticky tape, which you then can reach down and touch the hair. It'll stick to the tape, and then you can take the tape off and fold it over and record information on the tape, package it in an envelope or in a bindle, and it's preserved. You want to be careful to not be too aggressive with this taping, but sometimes you can do kind of a geographical taping. If you have like the trunk of a car, it may be important to know where in the car the hair was found. That may be able to help you determine whether the person was in a particular position, or perhaps you find something like body hair, and you can say, well, the individual was likely not fully clothed, so forth and so forth. So if you have an area like that, you can take strips of wide tape and lay them down side by side and label each strip. So then you put the tape down, you lift it back up, it picks up hair and fibers, which we're going to cover next, by the way. And then you can put it down on a piece of plexiglass in the same order that it was down in the trunk, and lay the plexiglass over a dark surface, which will let you see the light-colored fibers. Then lay it down on a light surface, which will let you see the dark fibers. And then, of course, you can search additionally with ALSs and so forth. And you may recall the little fiber demonstration I did when I brought the ALS to class, which seems like a long time ago. But anyway, that way you get an idea of the distribution of the hair, and you then proceed to see if that is something you can use or gives you some additional information. So collecting of samples for comparison is another issue. We, of course, if we have a hair from the scene of the crime, we'd like to have hair from the suspect and from the victim and from any other people who might have been in the area and who could possibly have been suspected or been involved. So first of all, we need to take the control. Now we're talking about a standard or a control. You need to take the hair from the same area of the body. If it's beard hair, we take beard. If it's scalp hair, we take scalp, etc., etc. And then don't just take one hair, because as I told you earlier, there's a range, there's a variation in one person's hair. Even mine, little bit of hair, you can see I have some darker hair and some almost white hair and some completely white hair. But we need to have the variation so that we can say, yeah, this person's hair is somewhere in this color range. So 25 hairs is kind of a good number. And of course, if we want to see the root and the characteristics in the base of the hair, we need to have that. So prefer to pull the hair rather than cut the hair. We'd like to get the whole hair. And there are exceptions to that. There are sometimes when it's not advisable to pluck the hair. For example, in sexual assault cases, rather than pluck pubic hair samples, we comb the area with a fine-tooth comb, which will accomplish two things, hopefully. One, it will give you standards which have been dormant and have come out or will come out in the combing process. So you'll have standards from the victim. But hopefully, if there's been a transfer of hair during the assault, then we'll have a sample of the suspect's hair as well. And so they recommend that you clip a sample of the victim's hair down, fairly close to the skin, and that then acts as an absolute standard. So we know that was a victim's hair because it was attached when we cut it. And so then we have samples in sexual assault kits, sexual assault investigation kits, I should say. We have a sample that's cutting and a sample that is a combing. The combing should include the comb, and so when you comb the hair, or have the nurse combs the hair, or whoever, have them keep the comb, put the comb and the hair from the comb in one package, and the clippings in another package. And the same is true for animals. Well, don't mean to relate animals to victims, but the same is true for collection of hair from animals. Take a good sample of the hair from different parts of the animal's body. And it's been shown by 2 or 3 studies that if you have a cat or a dog in your house, and someone comes into your house and spends 10 minutes, that they will in fact have hair from your animal on their clothing. So whenever you go to the scene of a crime and you see a pet, a dog or cat particularly, get a sample of them just in case. And that can easily be done. You don't have to hurt the dog or the cat. Just get a clean brush or a clean comb and comb their coat until you get a sufficient sample. So this about finishes up the hair section of our class. So I think we'll move on next. We're going to do fibers. So that'll be included in the same movie, only we'll just start out on fibers. About 40 years ago in Atlanta, Georgia, there was a series of murders of mostly young African American boys. The Atlanta police, the Georgia State Police, and the FBI began to investigate these murders because they seemed to be linked, and they were thinking perhaps they were committed by the same person. As part of this investigation, they began watching bodies of water because they, in their investigation, had learned that perhaps that was the next logical place to find a body. There was a policeman watching a bridge over the Chattahoochee River, and a car drove out on the bridge and stopped. And shortly thereafter, the police officer said he heard a splash, and then he saw a car drive away. The car was subsequently stopped, and it was determined that the driver of the car was a young man named Wayne Williams. Wayne Williams then became a prime suspect, and it was shown that he had association with some of the murder victims, lived close to them, or had some other kinds of social connection. Among the things that was discovered in the investigation was that some of the bodies of the murder victims contained fiber evidence, as well as some hair evidence. The hairs turned out to be dog hairs, and the fibers were of a couple of sources. One was a blanket, one was carpet, and one was carpet from a vehicle. The home carpet fibers were somewhat unique, and it took a thorough investigation to determine the source of those fibers. Turned out to be a relatively rare carpet fiber, only produced in small quantities, and hadn't been produced for a few years. The automobile carpet fibers were of a kind that is seen in other vehicles, of course, but not terribly common. The investigators then did two things which are almost unheard of. First of all, they determined the number of yards of carpet ought to be in the area of Atlanta, and then they sampled carpets and determined that this particular carpet was a rare one in Atlanta, that the odds of going into a room in Atlanta and finding this carpet were 1 in a few million. They then determined the same information for the automobiles. How many automobiles, how many of them had that kind of carpet, and what were the odds of finding that kind of carpet in a randomly selected car, which turned out to be 1 in a million or 2. So multiplying those together, because they're, as far as we know, those are independent variables. There's no direct relationship between the carpet in your car and the carpet in your house. It was determined that the odds of some person at random having both those kinds of carpets in their car and in their house was many, 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 many millions. So along with this and other information, they arrested Wayne Williams and charged him with the murder of 2 of the victims. Many people believed he was involved in others, but he was charged in the murder of 2. At the beginning of the trial, the defense attorney said, "You'll never convict him on a couple of fibers. What all I've got is a couple of fibers. What are we doing in a murder trial when we've only got a couple of fibers?" And it turns out that the evidence indicated that the likelihood of those fibers being found was so high and was presented so well that the jury convicted Wayne Williams of 2 murders. And he is still in prison, even though in the last couple of years there have been some exposés, if you will, that would try to lead us to believe that he's an innocent young man. Well, he's not young anymore. An innocent man. But it shows the power that can be derived from a careful examination of fiber evidence. There was, of course, other evidence besides fibers, but they were the best and most powerful evidence against Wayne Williams, because his car carpet and his home carpet matched in type and color the two samples, the several samples found on the bodies of the victims. So that's just kind of a brief introduction to fiber evidence. I've long thought that fiber evidence is one of the most overlooked kind of evidence. It's tedious to find. It's somewhat difficult to analyze. There are not a lot of fiber specialists. And even more importantly, rarely do we know what kind of fibers we're looking for, particularly if they're clothing fibers. They normally don't ask victims, well, what kind of shirt was he wearing? What kind of coat was he wearing? And so then when we go to the scenes of crimes, we don't know what we're looking for, so maybe we don't look. That's not recommending that procedure. I'm recommending you look always, but it's just human nature. If you don't know that there's any value in doing something, you tend not to do it. And oftentimes they think there's no value in fibers, when in fact there's a lot of value in fibers. There are some people who spend their lives investigating fiber evidence. There's a man in Montana who basically spent his career studying the transfer of fibers. So he has these little machines, and he mounts fibers on them, and then he rubs it together in a little machine, and he tries to determine what are the best conditions for fibers to be transferred from one article to another article. And he's done a lot of really good work. So we know now that when fibers are transferred, if you search for them in the first 24 hours, you've got a pretty good chance of finding transferred fibers on one or the other of the materials. After 24 hours, the likelihood goes way down. Well, that's good information to know. And it's not well known. I've heard him give several presentations at scientific meetings, and so I'm aware of his work and impressed by his his ability to stick with it. Well, okay, let's talk a little bit about fibers in general. Prior to middle of the 19th century, we basically had two kinds of fibers. We had natural fibers, and the natural fibers were mostly cotton and wool. There were different kinds of wool, and we'll discuss that briefly, but most of the clothing was made out of one or the other of those two fibers or a combination. Wool, of course, is the hair of an animal, typically sheep. It can be other animals' hair. There's camel wool, and there's alpaca wool, and there are various strains or breeds of sheep, but nevertheless it's a hair. So easily recognizable under the microscope as a hair, has all the characteristics that we just talked about in the hair section. And so we can very quickly determine whether a sample is wool. Cotton is a plant, the fibers of a plant, and cotton also has some microscopic characteristics that make it easy to identify. It's a long, narrow fiber that has twists. So if you look in a microscope and you've got a long, thin fiber with lots of twists in it, then it's almost certainly cotton. There are, of course, other plant materials from which fiber is made. Hemp, for example, sisal, and linen are all natural fibers that are derived from plants and are pretty easy to recognize microscopically. Around the 1940, 1950, somewhere in there, maybe a little earlier, we began to recognize the value of man-made fibers. The first ones were they would take a natural fiber or a natural growing plant that produces cellulose, which is the basic building block of lots of plants, particularly woody plants, and they could break the cellulose down and then extrude it through small holes called spinnerets and produce fibers such as rayon. And so those are man-made fibers. Then one, I don't know if he was lucky or brilliant, scientist discovered a way to create a polymer. A polymer is simply a long string of the similar molecular molecules. So it's just a whole big long string of the same kind of molecules. And he discovered this really nifty way of making polymers. And the story goes that when he made it, he first got a patent, and then he sent out letters to all the major chemical companies saying, "I have discovered a way to make inexpensive polymers, and I own the patent. I'm willing to make a deal." And he ended up getting $10 million and a penny a pound for everything made out of his polymer, which millions and millions of pounds. So he didn't ever have to go to work again. But anyway, he started this revolution in polymers, and polymer fibers, which we call synthetic because they're not made from plants or even from reconditioned or rearranged natural fibers. So synthetic fibers have become a major component of the fiber industry nowadays. And there are lots of them, lots of synthetic fibers. There's a chart in your book that takes up 2 pages that lists the names and strengths and weaknesses of various fibers. So we're not going to hold you to any of those things in the chart, but understand that synthetic fibers are ones that are totally made from man-made concoctions of chemicals, and they're polymers, and there's polyester, and there's nylon, and there's on and on and on and on. And so, since they're made in huge quantities in factories, and they're used in lots and lots of garments and so forth, there's very little variation from the format or the chemical composition of the fibers. Nevertheless, there's still things we can do to make comparisons, and we'll talk a little bit about those. The first examination for a fiber is microscopic. That is, you take a small sample of only a few of the fibers and look at them under a stereoscopic dissecting microscope at low power, which may reveal whether they are synthetic or regenerated or natural fibers. For example, you can often tell a hair under pretty low power because you can see the medulla and so forth. Then subject to higher power magnification under regular compound microscopes. So if you have a comparison, a hair and fiber comparison microscope, and you have a known and a question sample, you can compare them side by side at this point. If it turns out that they are natural fibers, then oftentimes the next step is to determine if the colors are similar, and we can do that very nicely under a stereo microscope. And the color and size of fibers is a very nice way of distinguishing them. Fibers, two fibers that are the same color and the same fiber, natural fiber, cotton, for example. There was a study done a number of years ago by a couple of criminalists in California to try to help figure out the value of or the probability of fibers coming from the same source. So they decided that the fiber they would look for is wool, and the color they would look for is blue. So they went out and searched high and low and found all the blue wool fibers they could find. And then they looked at them under the low power microscope and concluded with a little tricky mathematics that the odds of finding two blue wool fibers at random that matched was about 1 in 20. Now, and that, by the way, is where I got the 1 in 20 number to use in your assignment on fibers. That's not to say that that's a hard, fast number for all fibers by any means. It's that study, and that's the only one that we can say has a 1 in 20 chance. But if you've done the fiber assignment, you'll note that even with 1 in 20, if a person has 3 or 4 articles of clothing on, and almost all of us have, particularly in colder weather, then, if you've got 5 articles of clothing on, you've got a hat and a coat and a sweater and a jacket and pants and shoes or socks. So if each one of those fibers is odds are 1 in 20, then the combined odds, when you multiply them, the product is an enormous number. You come out very quickly with numbers of more than 1 in a million. So that's just an example of the value of microscopic comparison of color and fiber type. Now, the next step in that would be to do some kind of a chemical analysis on the dye. Most fibers are dyed nowadays. Most clothing fibers are dyed. And if it's cotton, there's certainly a huge percentage of cotton fibers that are white. But there's also lots of other colors. And comparing them under the microscope, if you can see a difference in the color, then okay, you found that they don't match. And your eye, as we've talked about earlier, is pretty good at seeing colors. If they look the same, they still may be slightly different, and so you need to do some kind of chemical analysis. And what is commonly done is you take a fiber and insert it in a small, what we call a capillary tube, which is a tube about the size of a pencil lead, and they're used a lot in medicine, but also in chemical analysis. So you put a fiber in there, and then close off one end with heat, a Bunsen burner or match or whatever, and put some solvent in the capillary tube with the fiber. Seal off the other end and put it under a low temperature, about 70 degrees Celsius or so, overnight, which will then dissolve the dyes out of the fiber. You come back in the next morning, and you can do thin layer chromatography on that the solvent in the tube, or gas chromatography. And of course, gas chromatography is more distinguishing than thin layer. But in either event, you've got a chemical analysis, and you can also take that liquid and spot it on a plate and do spectrophotometry. We now have micro spectrophotometers, which allow you to examine something under the microscope and then analyze that beam of light that's gone through the sample in the microscope from the sample up to your eye. They just take that same light and give you an infrared spectrum. So you've got an infrared spectrum, gas chromatograph, retention times, your eye, and combine those together, you've got a pretty powerful comparison protocol for natural fibers. In synthetic fibers, we would go through basically the same process, but we now want to know what kind of fiber is it as well. Before we knew it was cotton or we knew it was wool, but in synthetic fibers we have to determine that, and probably the most common way to determine that is infrared spectrophotometry. So we do an infrared spectrum of the sample and compare that to spectra of the known synthetics. We also look at the characteristics, physical characteristics of the fiber, particularly cross section. So some of them are round, and some of them are triangular, and some of them are barbell shaped, and so there's numerous shapes for these fibers, and it depends on the spinneret that they went through, what the shape is. But some of them are more common in one shape than others. So the physical characteristics combined with the infrared spectrum will allow you to identify the name of the particular fiber. That, you know, is it polyester? Is it nylon? Is it, you know, whatever it is. There, by the way, there's not just one nylon. There's 6 or 7 different nylons, and nobody knows all the applications for polyesters. It seems like you can find a polyester to do just about whatever you want. In any event, then you have, of course, the same system for identifying the dyes that you had with the natural fibers, that is, extracting them in a capillary tube and running spectrophotometry and gas chromatography. So those are the analytical tools for comparing fibers. Occasionally we get a piece of fabric, which is, of course, fibers made into threads, threads made into cloth. And so we have all of those characteristics we can compare. So we can look at particular fibers, but we can then look at the threads and say, okay, we have this particular thread, it's this diameter, it has this many twists per inch or per centimeter. and the twists go in the right-hand direction or the left-hand direction. And so we have those kinds of physical characteristics that we can use when we have a section of cloth. We can do the same thing with rope. You have a rope, and if it's a twisted rope, it'll usually have more than one ply. So how many plies does it have? How many twists per inch in each ply, and so forth and so forth. So we can do these kinds of physical comparisons, which obviously are helpful. They don't individualize, but fiber evidence is not the type of evidence that lends itself to individualization. And the question is, okay, so we've got a fiber, and we know it's from a shirt or from a piece of cloth like a shirt, and the fiber matches a fiber found at the scene of the crime. So what's the probability that it's from this victim's shirt and not another shirt just like it? And we don't have any good way of tracing. It would be nice if we had those kinds of numbers and we knew, okay, there's only this many of this kind of shirt around, and then we could do some mathematical manipulation. But we don't, and so we just have to rely on experience. Sometimes you can, if it's a rare fiber like the one in the Wayne Williams case, then you may be able to do some additional probability studies. But by and large, what we're left with is the experience of the examiner and maybe the probability of contacting companies and find out, okay, how many of these kinds of shirts did you order at your store, and hopefully you're the only store that ordered them. But it is just, I think, not good practice for an examiner to try to indicate that this is individual evidence. This is class characteristic type evidence, but still very powerful, as Wayne Williams would admit, among many others who've been convicted as a result of fiber comparisons. What I said in the video on hairs in regard to collection preservation is pretty much the same for fibers. We have to be very careful, very tedious, and very patient in collecting fibers. They're often very small, and you need to use at least a magnifying glass to find them most times. And they need to be collected carefully and made sure that they're not contaminated by fibers from another source or that might be floating in the air. The big thing is crime labs and crime lab personnel need to be informed if there's some kind of possibility that fiber evidence might be important, and that will alert them to be on the lookout as they examine victims and suspects' clothing and so forth. The use of ALS and similar lights is encouraged, and sometimes it is pretty dramatic, as it was in the case of those fluorescing orange fibers on my sweater back when we had that class. But they need to be properly collected, documented, photographed when possible, and packaged so they won't get destroyed, of course. If you have clothing, then always be sure that it is carefully folded, not stuffed into a bag. And if there are areas areas of importance, such as you see some fibers, then you should protect that area by putting pieces of paper between the folds so that the evidence won't get transferred from one part of the clothing to another, which is just standard practice, but isn't always followed. It is usually in the case of blood, but not often in the case of fibers. But these are the things that we need to train the crime scene investigators, because I'm absolutely convinced that a lot of fiber evidence is overlooked, and we could solve crimes if we were more careful searching for fibers.